Okay, Assalamu alaikum. Welcome again. So, as I said that yesterday we were discussing about the process of DNA replication, and there were a couple of things that we discussed. So, I'm going to continue from where we ended up yesterday. We were discussing about DNA replication in particular reference to bacteria or prokaryotes. And in generally speaking, the process of the DNA replication, no matter it's in pro or eukaryotes, is itself a complicated process because it involves many enzymes, it involves many proteins, and it involves many steps that occurs side by side or simultaneously. Out of all these, the key enzyme, the main enzyme that is involved in the process of replication are the DNA polymerases. Now, these are the enzymes which catalyze the joining reaction of nucleotides with the phosphodiester linkage. And as a result of that, the nucleotides will be add on and there will be a formation of a new polynucleotide chain, which is definitely compatible and comparable to the parental chain, which already exists. So the parental chain is working like a template, and a new polynucleotide chain is being synthesized considering the template sequence, and that reaction is catalyzed by DNA polymerase enzymes. Yesterday, we studied about the fact that it's not just the DNA polymerase starts the reaction, but there are few steps and there are few enzymes which are key towards the process of initiation of the DNA replication process. Once the process is initiated, then the DNA polymerase can work to add on the nucleotides to grow the chain. So one thing which we discussed yesterday is the DNA replication is always carried on in the direction of 5 prime to 3 prime. So this point is very important and that has to be kept in mind. Then always the newly synthesized chain of DNA strand must go through in the direction of 5 prime to 3 prime. So the template that it's gonna copy is in the orientation of 3 prime to 5 prime. So when the template itself is 3 prime to 5 prime, the newly synthesized chain will be in the direction of 5 prime to 3 prime. Now, the point is that when we talk about the DNA molecule, as I repeatedly said, that it doesn't matter that if it's a circular DNA of a prokaryote or it's a linear DNA of the eukaryote, each of the DNA molecule is a double-stranded molecule. It's just about how it is arranged. So in prokaryotes, the double-stranded DNA is in a circular form, and in the eukaryote, the double-stranded DNA is linear. So we have two strands which are, bonded together with the hydrogen bonding. So the point is that if one of the strand of the template DNA is 3 prime to 5 prime, there should be the other strand that will be having the opposite orientation, which is 5 prime to 3 prime. So as I said earlier on, that a new strand can only be synthesized in the orientation of 5 prime to 3 prime. So what it reflects that out of the DNA molecule, which is the parental DNA molecule, one of the strand of the parental DNA, which is 3 prime to 5 prime, it's quite simple to synthesize something on it which is 5 prime to 3 prime. But what about the second parental strand? The second parental strand itself is having an orientation which is 5 prime to 3 prime. So how a new strand can be synthesized on that strand? This is uh, an important question. So before we proceed towards this, let me tell you that the parental strand, which is having 3 prime to 5 prime orientation, is called the leading strand. And that leading strand is very simple because The orientation of the leading strand is itself 3' to 5', so a new strand can be easily synthesized on that with a 5' to 3' orientation. What about the other one, as I said, the other parental strand, which itself is running 5' to 3', it's being referred as lagging strand. Now, for the lagging strand, there is an issue because the lagging strand has an orientation of 5' to 3'. So how is it possible to synthesize a new strand by using lagging strand as a template, because lagging strand already has the 5' to 3' orientation, and you cannot synthesize the new strand by the orientation of 5' to 3' because they both are the same. And as I mentioned earlier, that the newly synthesized strand must always be carried on in 5' to 3'. So the answer to this question is that on the lagging strand, which is the second part of the parental DNA strand, which already has 5' to 3' orientation, the newly synthesized chain will be synthesized in small fragments or in the form of discontinuously short fragments, and that will be explained to you later on when we go in the details. We'll see how it carries on. So just to make sure that out of the parental DNA molecule, one strand is called leading strand, whose orientation is 3' to 5'. The other strand is the lagging strand. The orientation for that is 5' to 3'. Now, for the leading strand, there is no issue at all because leading strand is 3' to 5', and we can easily synthesize a new chain on this, which runs in 5' to 3', which is opposite in direction. But the problem comes with the lagging strand because lagging strand is already 5' to 3'. And how we going to synthesize a new chain by using the lagging strand, which must be going in 5' to 3'? So that's the question, and that we're going to discuss later on. Some of the reflections are there that what happens actually that on the lagging strand, we do not have a continuous chain formation. Rather, we have short fragments of the nucleotides synthesized on the lagging strand. And once these short fragments are synthesized, all of them are joined together to make the complete chain. These short fragments, which are being synthesized on the lagging strand, are called as Okazaki fragments. Okazaki was the name of a Japanese scientist who discovered this process, and on that basis, we call those fragments as Okazaki fragments. These Okazaki fragments, they are ranging from 100 to 1000 nucleotides long. So when the synthesis is going on the lagging strand, using as a template, we have small fragments of 100 to 1000 nucleotides synthesized sequentially. And then once this process is completed, these fragments are joined together. These fragments will be connected together to make a whole complete new chain. So that's why there is a difference when it comes to the synthesis of a new strand on the leading strand and on the lagging strand. Okay, so as I said, that DNA polymerase is the key enzyme, and I mentioned that point that what DNA polymerase is doing. DNA polymerase is catalyzing the reaction of joining of two nucleotides via phosphodiester linkage, and that's how the chain elongation takes place. But if you remember, we studied yesterday that DNA polymerase cannot directly start the process. There are few things that must be considered before the DNA polymerase comes into action. Now, what are these things? We will go again in a little bit of detail. So what we say that the DNA polymerase enzyme cannot begin a new DNA chain from the scratch. This is one thing. This is a very important point. The polymerase enzyme of the prokaryotes doesn't have this property to synthesize the DNA chain right from the scratch. What does that mean? It means that the DNA polymerase cannot join two free nucleotides to make a dinucleotide. And then by using the dinucleotide, add another one, trinucleotide, add another one, and that makes a polynucleotide. It doesn't work like this. So DNA polymerase cannot start the nucleotide addition right from the beginning. So I repeat, the DNA polymerase cannot join two free nucleotides to make a dinucleotide. This is a very, very important point when it comes to the DNA polymerase in bacteria. So what is the solution? It can only attach a new nucleotide on the 3' end of a pre-existing strand. What does that mean? The DNA polymerase enzyme requires a small pre-existing strand of nucleotide upon which it can add on one nucleotide. So you got my point? The DNA polymerase cannot join two nucleotides right from the beginning. Rather, it requires a pre-existing strand with a few of the nucleotides already attached together. And with that pre-existing strand, the DNA polymerase is capable of adding up the new nucleotide on the 3' hydroxyl group of this pre-existing strand. This pre-existing strand, which is a must condition for this DNA polymerase in bacteria to add on the new nucleotide, is termed as primer. This pre-existing strand is called primer. So that means we need a primer. We need an initiator. We need a pre-existing small nucleotide chain upon which the DNA polymerase can add on the further nucleotides. So in this case, to start the synthesis of the leading strand, each DNA fragment of the lagging strand, an RNA polymerase complex called a primosome or primase is required. Now, coming back to the point which I said that before the DNA replication, we know that the first step was unwinding because these two strands needs to be separated out. There are some other conditions. As I said, that DNA polymerase cannot directly start. So we have another enzyme called RNA polymerase. Now that enzyme is very much similar in its role to the DNA polymerase, but it's called RNA polymerase because what it is going to do, it's going to synthesize a small fragment of the nucleotide, which I'm saying as primer. So RNA polymerase is the one which is playing an important role in this case. Similarly, we have another enzyme called primase, which is the main enzyme synthesizing the primer. So it's bit of a complicated structure where you got the lagging strand, you got the RNA polymerase enzyme, you got primase enzyme, and this collectively being called as primosome. Now, the primase is, as I said, is the enzyme which is capable of joining the RNA nucleotides without requiring a pre-existing strand of the nucleic acid. That what forms what is called RNA primer. So, going back to the point, the DNA polymerase is not capable of adding on the nucleotides right from the beginning. They need a small set of the nucleotides in order to carry on the reaction. That small set of the nucleotides required is called primer, or you can say RNA primer. Why we call it RNA primer? Because it's a single strand. That's why we call it RNA. This RNA primer is actually being synthesized by the enzyme primase. And this primase can make a small pre-existing strand, and that primer can be used by the DNA polymerase to add on further nucleotides to carry on the DNA replication. So look at the picture here. You can see that this is called the replication fork. The two strands have been separated. This purple color picture is of RNA primer. And you can see that on this RNA primer, we have the chain synthesis going on. This green one is a newly synthesized DNA molecule. So one is going in this direction and one is going in this direction. So this is actually the new DNA molecule. And this RNA primer is being synthesized by primase, which is essential part to begin the process of the DNA replication. We studied yesterday that the DNA replication fork, it goes bidirectional. So you can see that one going in this direction and one going in this direction. And as they go in these opposite directions, the unwinding carries on and the new chains are being simultaneously synthesized, as you can see in the picture. Now, after a few nucleotides are added, primer is complete. So once the primer is ready, the primase will be replaced by DNA polymerase, because the purpose of the primase enzyme is to make a primer. So once the primer is there, now the primase will be removed and it's being replaced by the actual DNA polymerase enzyme. And now this DNA polymerase will do its function. It will add on a free nucleotide to the 3' end of this short RNA primer. Once this whole process of DNA replication is complete, this primer, which was synthesized in the first step, will be removed and degraded. Why? Because that primer is not the actual part of the DNA. Don't forget that this primer has been synthesized. Why? Because DNA polymerase enzyme lacks the capacity to join two free nucleotides. So in this case, make sure that this primer has been made just to facilitate the DNA polymerase to synthesize the DNA molecule. So itself, the primer is not the part of the DNA structure. So once the DNA molecule or the chain has been synthesized, this primer is no more needed. So it's gonna cut down from the chain and it's degraded, and that's how the DNA replication process is completed. Now in bacteria, there are different kinds of DNA polymerases, and I'm going to explain or give some information about few of them with their specific functions. The first one is the polymerase 1. This DNA polymerase 1 has a specific function of DNA repair. It has the capacity to do the polymerase activity, which is the main activity, in 5' to 3' direction. Remember, I repeatedly said that the addition of the new nucleotide chain will always be in the direction of 5' to 3'. So I can say that the second function of this polymerase is actually the main function of any DNA polymerase. Any DNA polymerase would have this function. But on top of this, this DNA polymerase 1 has a function of DNA repair as well. It also has another function, which is a proofreading function. What is proofread? I think the name itself is self-explanatory. Once the DNA chain is being synthesized, we need to check that whether the correct nucleotide sequences have been put into practice. Otherwise, if it's not the correct nucleotide, we know that it's a mutation. And as a result of the mutation, there will be a wrong product, wrong protein. So in this case, we are not only interested in DNA replication, but we also are interested in the proofreading. So this DNA polymerase 1, on top of its polymerase activity, is also having the property of the proofreading. And not only the proofreading, but if it finds a wrong nucleotide which is accidentally being added in the DNA chain, it can remove that. And this is called 5' to 3' exonuclease activity. And it is also removing the RNA primer, which is being synthesized, earlier on, to provide a pre-existing chain for the DNA polymerase to start work. So polymerase 1 has a function of repair. It has a function which is the basic function of any DNA polymerase, which is 5' to 3' polymerase activity. It has a function of proofreading in the direction of 3' to 5'. It has a function of 5' to 3' exonuclease activity, and it also removes RNA primer once the process of the replication is complete. So DNA polymerase 1 is actually not the main enzyme, I would say, not the main enzyme that do the replicative polymerase, because of the following factors. Number one, this enzyme is too slow, like the speed with which it catalyzes the reaction is very slow. You can see that it adds the DNTPs. DNTP is the free nucleotides at a rate of 20 NT per second. So with this speed of 20 nucleotide per second, so it would require about 460,000 seconds. Or if you cut down this to the days, it will be 5.3 days to replicate the E. coli chromosome. So you can better understand that the E. coli chromosome is the simplest of the chromosome because it's a bacteria. So if DNA polymerase 1 is the main enzyme doing the DNA replication, it requires 5.3 days to replicate the entire genetic material of E. coli, which is a very, very long time. And that's why we say it's too slow for an organism because the E. coli can divide every 20 minutes. So that means this DNA polymerase 1 is not doing the main function of polymerase activity, which is, catalyzing the chain formation. But it does have other functions, as I said, proofread and, DNA repair and so on and so on. The second reason why it's not the main enzyme is that this enzyme is too abundant. There are 400 molecules per E. coli cell, and this is excessive given that there are generally only two replication forks in a cell. The third reason is the enzyme is not processive enough. What does that mean? It means that the DNA polymerase 1 dissociate after catalyzing the incorporation of 20 to 50 nucleotides. So it doesn't have a tendency to catalyze long reaction. It can only add on up to 20 to 50 nucleotides after which it dissociate. So it cannot carry long chain formation. So what we summarize, the DNA polymerase 1 does have the polymerase activity, but it's not the main enzyme for the DNA replication in E. coli. The second kind is the polymerase 2. It has also two functions. Number one, it is involved in the repair of the damaged DNA, just like polymerase 1. And it also has 3' to 5' exonuclease activity, which is a kind of proofreading activity. So that proves that this is not the main polymerase as well, because you can see that we haven't mentioned that this enzyme is doing the addition of the chain. So polymerase 1, neither, and polymerase 2, they are not the main DNA polymerase enzyme which are involved in the new chain formation. And the proof that why it's not the main polymerase, now again there are 3 points. Number one, the E. coli strains lacking the gene show no defect in growth or replication. That means there are certain E. coli, they don't have DNA polymerase 2. And in absence of the DNA polymerase 2, there is no problem with their replication. That means this enzyme is not the key enzyme for replication. Similarly, synthesis of polymerase 2 is induced during stationary phase of the cell growth. If you remember, I mentioned you the cycles of the cell growth. A phase in which little growth and DNA synthesis occurs. So that's why this is not the main enzyme. And third point is the polymerase 2 has a low error rate, but it is much too slow, just like polymerase 1, to be used for DNA synthesis. So these are the points why DNA polymerase 2 is not the main enzyme doing the new strand synthesis. Now here comes the DNA polymerase 3, which is in fact the main polymerase enzyme doing the DNA replication in bacteria. So DNA polymerase 3 is the main polymerase enzyme which is specifically involved in the elongation of the new chain, or in other words, involved in DNA replication. It has 3' to 5' exonuclease proofreading ability as well. So what I summarize, polymerase 1, 2, 3, they all have proofreading mechanism. Polymerase 1 and 2 are also having additional mechanism of DNA repair, but they do not have the main function of DNA replication, while this DNA polymerase 3 is the main enzyme which is involved in the DNA replication. So we said that it is the principal replicative enzyme. And how we can prove that? There are two reasons. Its processivity is very high. Remember, in polymerase 1, the processivity was 20 to 50 nucleotide, after which it dissociates. But for that, The processivity is very high. It carries on thousands and thousands of reactions. And the second proof is that the reaction rate is very high. It catalyzes the reaction very quickly. If you remember, I just told you that for polymerase 1, it roughly requires 5 days to complete the replication of the E. coli genetic material, while the E. coli roughly replicates or doubles in 20 minutes. So that means the DNA polymerase 1 is not doing the replication, but DNA polymerase 3 has got a very, very high catalytic rate. Now, we have gone through DNA polymerase 1, 2, and 3 in detail, and we also have seen their differences. Going further, the enzymes have got two parts which we need to understand, and this is you have already studied in your 312. The two forms of the enzymes are core enzyme and hollow enzyme. Now, what's the function? The core enzyme is actually the one that consists of the subunits that are required for the basic enzyme activity. Like, any enzyme has got a basic enzyme activity, or in other words, a reaction which it catalyzes. So the core enzyme is that part of the enzyme which has only those subunits which are integral part of its enzyme activity, which are integral part of its enzyme catalyzing reaction, like alpha, delta, theta, epsilon, etc. These are the names of small subunits which make up the core enzyme. Beside the core enzyme, we have got a hollow enzyme. What is hollow enzyme? The hollow enzyme is the fully functional form of an enzyme. What does that mean? It complete with all of its necessary accessory subunits. So that means there are certain subunits which are the basic subunits catalyzing the reaction, but there are some accessory parts which are also required for the overall enzyme activity. So once this accessory subunits combine with the essential subunits, that's called the hollow enzyme. So the DNA polymerase 3 hollow enzyme consists of the core enzyme and the sliding clamp, which is the accessory part, as I said, and a clamp loading complex. So when these, the accessory part and the essential parts combine together to make a bigger molecule, that's called hollow enzyme. So in short, we can say that hollow enzyme is the complete, fully functional form of an enzyme. The core enzyme is the actual enzyme which is involved in the catalytic reaction, but the core enzyme is not capable of catalyzing the reaction until unless it has got the accessories with it. So once the accessories are there, it becomes hollow enzyme. Now there are two more polymerases. I'll quickly go through that. Polymerase 4 and polymerase 5. The polymerase 4 and 5 are from the Y family of DNA polymerases, and their main function is bypassing the DNA damage role. Now, this is a picture again. That's a complete picture of the DNA replication. I'll spend some minutes on this picture to give you a bit of the overview of what we have studied till now. So if you look at this strand, this one, this is the leading strand, and that is the lagging strand. On the leading strand and the lagging strand, this is the point from where they, you can see the replication fork is there and the unwinding takes place. So as the unwinding takes place, this leading strand is 3' to 5', and the chain which is being synthesized on that is 5' to 3'. And you can see that this is a continuous chain formation. The enzyme which is mainly responsible in this, as I just mentioned, is DNA polymerase 3. Before DNA polymerase 3, we must remember few steps. Number one, unwinding, which is being done by helicases. I mentioned you yesterday. Once there is an unwinding, there is a topological stress I mentioned, which is being removed by topoisomerases. Third point, there were certain proteins which bind with the free strand and prevent the reunion of the two chains. So these are the three very, very important points. The fourth point, which I mentioned today, is the DNA polymerase cannot start from the very beginning. So we need a small primer, and this primer is being synthesized by primase, which is RNA polymerase. And the last point is that once the DNA chain synthesis is complete, the primer is no more needed and it has to be removed because that's not the part of the DNA itself. Now, coming back to the lagging strand, you can see for the lagging strand, the orientation is 5 prime to 3 prime. Now, in this case of the 5 prime to 3 prime, as I mentioned that there is no issue when it comes to the leading strand because there is a continuous synthesis going on. But with this case of the 5 prime to 3 prime, we cannot have the continuous synthesis because if the continuous synthesis starts from there, it will be again 3 prime to 5 prime, which is wrong because we said the DNA chain formation cannot be done by 5 prime to 3 prime. Sorry, must be done by 5 prime to 3 prime. It cannot be from 3 prime to 5 prime. So you can see that there are short fragments, these brown arrows. They are being synthesized upon this template strand. This is the first fragment, second fragment, third fragment, fourth fragment, and fifth fragment. And you can see the direction of them is 5 prime to 3 prime, 5 prime to 3 prime. So these small fragments which are being synthesized, they are called the Okazaki fragments, as I mentioned earlier. And once this whole chain synthesis is done, these Okazaki fragments will combine together and will make a complete chain. Which enzyme is going to join these fragments? This enzyme is called DNA ligase, which will join the fragments on the lagging strand once the synthesis is done. Another very important point to mention here that in this case of the leading strand, you need only one primer. Once the primer is there, now the DNA polymerase starts its function. But on the lagging strand, because the chain formation is not continuous, we have got small fragments. So for every fragment, you need a primer. So you can see here there are more RNA polymerases being shown here with this blue color. What they are doing, they are preparing primer. So there are multiple primers needed for the lagging strand, and there is only one primer needed for the leading strand. This is another very, very important point to be make sure of. This is another animation picture. Uh, Today I've just checked that a few of these might not be working. Maybe they are old enough or they have been taken out from the website. But you know, you can search on the YouTube and you can see many 3D animated pictures which can explain you that how this 3D picture looks like when you look at the replication process, and it will definitely help you a lot to understand because these sort of pictures are pretty difficult, but for the 3D videos, it's much easier to understand. So you can search on the Google and on the YouTube. I'll also try to search more if I found something. I'm going to share with you guys. So I think we still have some time, but I don't want to, as I said, complicate the thing. So I'm going to stop here because we have completed DNA replication in prokaryotes. So I want you guys to go through the slides again and again and come up with a question if you have questions in order to fully understand this process. Once you are aware of that, the next week in the class we will be starting up with the DNA replication in eukaryotes, because of course the DNA replication in eukaryotes will be a little bit more complicated than what you have studied in the prokaryotes, and that makes sense. So we are going to stop here, and I'm going to ask you that if you have any question, just raise your hand. I'm available for answers. Yes, Nimra, go ahead.
अस्सलाम वालेकुम सर।
जी वालेकुम अस्सलाम।
सर आपने बताया है कि पॉलीमरेज़ 2 में जो एक्सोन्यूक्लियेसेस होता है जिससे जो लाइक जो न्यूक्लियोटाइड्स जो ठीक सीक्वेंस से अटैच नहीं होते वो रिमूव होते हैं। तो
जी
पॉलीमरेज़ 2 में 5 प्राइम टू 3 प्राइम हो रहा है और पॉलीमरेज 3 में 2 प्राइम टू सॉरी 3 प्राइम टू 5 लाइक ऑल्टरनेट हो रहा है इन दोनों का। तो आप वो प्रोसीजर बता सकते हैं कि किस तरह से होता है कि एक में चेंज है और दूसरे में चेंज है?
Okay, let me explain you. I think there might be a confusion. दो चीज़ों को जहन में क्लियर रखिएगा. Whenever there is addition of nucleotides going on, चाहे वो लीडिंग पे हो रहा हो, चाहे वो लैगिंग पे हो रहा हो, चाहे वो डीएनए पॉलीमरेज 1 2 3 4 5 में से कोई भी कर रहा हो, एडिशन ऑफ न्यूक्लियोटाइड विल ऑलवेज बी 5 प्राइम टू 3 प्राइम. ये थंब रूल है. कभी भी इसके अलावा नहीं हो सकता. जब भी ऐड होगा न्यूक्लियोटाइड, इट्स ऑलवेज 5 प्राइम टू 3 प्राइम. जो एक्सोन्यूक्लियस की हम बात कर रहे हैं, दैट इज ऑलवेज 3 प्राइम टू 5 प्राइम, बिकॉज जब आप रिमूव कर रहे होंगे किसी न्यूक्लियोटाइड को तो उसका ओरिएंटेशन ऑपोजिट होगा. तो जो भी डीएनए पॉलीमरेजेस प्रूफ रीडिंग में इन्वॉल्व होते हैं, they always have this tendency of 3 prime to 5 prime exonucleus, because exo का मतलब है removal. So whenever a nucleotide is being removed, it is always 3 prime to 5 prime, and whenever a nucleotide is being added, it is always 5 prime to 3 prime. I hope that answers the question.
So sir, it will be same for polymerase 2 and 3 as from 3 prime
Yes,
to 5 prime.
yes. Any exonucleus activity by any of the polymerases will always be 3 prime to 5 prime.
Thank you sir.
Welcome. Any more questions, guys? Yes, Uruj, carry on.
Assalamualaikum, sir. Can you please again repeat? Addition is always 3 to 5 prime, right?
No. Addition is always 5 prime to 3 prime.
Yeah,
Removal is
sorry, it's 5
always
prime.
3 prime. Yeah. Removal
Okay,
is
just
always
wanted to
3
hear.
prime to 5 prime. Yeah.
Thank you, sir.
Welcome. Any more question, guys? If you don't have question at this stage, I will highly recommend you to go through the presentation, because I have already shared the presentation and lecture is already there. So go through it. And if you have got any question, do come up with the question in the next class, or meanwhile you can ask me directly whenever you want to ask the question. So with this, we will stop the class. Thank you very much, and inshallah, we'll see you in the lab on Monday, and I'll consider with talking to your CR regarding the timing. Thank you very much. Khuda Hafiz.